Good afternoon, students.
Good afternoon, sir. Good afternoon,
Just let
sir.
me let me just set my camera. I think there is some issue. Okay, students. So by the time I set the camera, so now we have talked about uh, antibodies, right? So now you know the structure of antibody, you know the different types of antibodies, and you also know the role of different types of antibody, right? Because there are different types of antibodies and nature has given different role to different antibody, right? Like one of the antibody, it can be in the external secretions, it can be in the mucus and protection, right? Okay, so now the camera is working. So another one, it will help in protection against parasites, okay? Because to kill a parasite, you need to have some different mechanism, right? I think just understand, a bacteria is very small, a virus is very small, right? So a cell can come and engulf it, right? But a parasite may be bigger than a macrophage. So obviously this macrophage will not be able to engulf it. Okay. So you need a different mechanism. So there IgE comes and play an important role. Right? The baby, the fetus also needs some protection. So there are some antibody types which can cross the placenta, right? So different types of antibodies, they have different type of roles, and we have already seen that. We know the structure, they are different, right? So today we will talk about monoclonal antibodies. Okay, so what we have learned till now, we have talked about antibodies in context to what we see in the body. Okay, but scientists have now utilizing these antibodies for various purpose. Okay, scientists have been able to modify these antibodies, or they have been made selective or specific antibodies, right? A lot of changes have been made in these antibodies so that they can be used for various purpose, right? One of the example is diagnostics. Okay, so just for example, just hold on. Okay, so as I said, one of the example is diagnostics, right? So तुम्हें कोई भी diagnosis करना है, for example of COVID. I'm sure you all know that rapid antigen test, right? So whenever we are doing some diagnostic test, there is antigen and it has to bind with an antibody, right? So I'm sure you have read about ELISA. Have you read about ELISA in any other subject?
Yes, sir, we have.
Yes, so you know what happens in ELISA. You have an antibody, right? And then the antigen binds to that antibody, right? So there are different types. Either it could be a sandwich in which you have antibody to which the antigen bind, or you can just put the antigen to which the antibody can bind, right? So there are different modifications of ELISA. So for all those things, you need specific antibodies, right? You need monoclonal antibodies. So today we are going to talk about monoclonal antibodies, how they are developed, okay? And maybe in the next class, we will talk about the applications of these monoclonal antibodies. So there are a lot of different applications. I've just discussed one of the application, right? And when we talk about diagnostics, it's a very big field. Okay, so we'll talk about that. We'll talk about the applications. But before we move on to monoclonal antibodies, let's refresh uh, through a quiz, right? What we have learned about the different classes of antibodies. Okay, so there is a quiz on uh, different types of antibodies. So if everything is clear there, okay, then we will move ahead to the next topic, which is monoclonal antibodies. Because if you understand the structure of antibodies well, if you understand how these antibodies work, then you will better understand monoclonal antibodies. Okay, so I'll start with the quiz. So let me just assign it to the class and then it will be visible at your end. Okay, students, so I have assigned it, okay, and I'm sure it is now visible at your end. Yes, it should be visible at your end now. Is it visible now?
Yes, sir.
Okay, students. So quickly, just give the answers, okay? And then we'll just discuss the answers and we'll move ahead to our next topic, which is monoclonal antibodies, okay? And this is a very interesting topic, monoclonal antibodies. And when you will read about its applications, you will be amazed how we are making use of these antibodies for various purposes. Okay, so just quickly do the MCQs, give your answers, just do it quickly. So students, start submitting now. I can see 3 students have submitted. So rest of you start submitting. Students, start submitting now. You've already taken so much time. Not even half the class has submitted. Do it fast. So students, just 4 or 5 students are left. Do it fast. Students, do it fast. Last 2 minutes. Okay, students, so uh, let's discuss the answers. Okay, so the first question was immunoglobulin G is subdivided further on the basis of. So, 5 students say that it is because of the size of hinge region. 3 students says it's because of is on the basis of number of interchain disulfide bond. Okay, but the correct answer is all of the above. Okay, so IgG is subdivided into 4 types on the basis of the size of the hinge region. Some types have bigger hinge region as compared to others. Number of interchain disulfide bonds. So I hope you remember that picture. Some had 2, some had 4, some had 11. So number of disulfide bonds and the position of disulfide bond. You remember some were below the hinge region, some were above the hinge region. So uh, on the basis of that and uh, so the correct answer is all of the above. Okay, so the correct answer is all of the above. Okay, question 2, which is the first antibody type that interact with the antigen? Only 2 students have given wrong answer. The correct answer is IgM. Okay, which antibody is present in external secretions? One student has given wrong answer. Rest all of you have given the right answer. It is IgA. Okay. Question 4 was how many CH domains are there in IgE on light chain? Okay, so uh, 2 students have given wrong answer. They have mentioned 4, and I don't know how they have come to that number, but the correct answer is 2 because light chain only have 2 domain. One is the variable region domain and other one is the constant region domain, right? So if you see one molecule, there is 2 light chain, okay? So both the light chain will have one uh, constant region domain. So total of 2 constant region domain. Okay. Question 5 was which is not true with regard to hinge region. Okay. Now 4 students have given wrong answer. The correct answer is it is rich in cysteine and arginine. Okay. So it is not rich in cysteine and arginine. It is rich in cysteine and proline. Okay. So fifth ka correct answer is it is rich in cysteine and arginine, which is a wrong statement. Okay. Question 6 was how many subclasses does IgG and IgA have? Okay. Again, I can see 4 students have given wrong answers, so I don't know why the number is high. It was very simple. IgG have 4 subclasses and IgA have 2 subclasses. The correct answer is 4 and 2, right? So I don't know. Again, question 7. One student has given wrong answer. Which of the following pair do not have the hinge region? Correct answer is IgE, IgM. Okay, because uh, IgE and IgM does not, they have an extra constant region domain, so they don't have the hinge region, right? So they have 4 constant region domains in their heavy chain. So the correct answer is IgE and IgM. They do not have the hinge region. Okay. So with this, uh, okay. So if I just take the average score of the class, so out of 16, the average score is 13.5. Okay. So uh, you just need to read it again. Okay. There are a lot of minute things that you need to uh, just take care of. Okay. Okay, so let's start with the topic of today, which is monoclonal antibody. Okay, so let me just share the slides. Okay. So students will be talking about monoclonal antibodies, and you can see like the blue image. Suppose it's an antigen. What happens? You have the antibodies, they come and bind like this. So let's talk about now monoclonal antibodies. Okay. Now, monoclonal antibodies, they are effective in vitro. Okay, they are used for research, diagnostic, and therapeutic purpose. Okay, now the most important thing about monoclonal antibody is that it is derived from single clone, right? Specific for a single epitope. Okay, so monoclonal antibodies are specific against a specific epitope, right? So you can see here, this is a particular cell, okay, and this cell have a lot of different proteins or epitopes. So right, you can see there are different epitopes. Just for example, this blue epitope, this green epitope. So basically different proteins, right? So one cell can have many different epitopes, okay, and monoclonal antibody will get locked onto a specific epitope, right? So this will only bind to this triangular epitope, okay. So monoclonal antibodies are specific against specific epitopes, okay. Now as I said, they have a lot of applications, right? So there was a need to develop monoclonal antibodies, right? Suppose you have to make a ELISA kit, right? So for that, if you have to make it thousands of ELISA kits, right? You also need a large amount of monoclonal antibodies, right? So one of the method is you inject a mice, okay, or you inject a rabbit, you inject some animal, okay, and obviously in that animal you will find specific antibodies. Then you can take out its blood, purify the antibodies and use it, right? So that is one method. But you can just understand how complicated it is because you need a large animal house. You will need a lot of animals for that purpose, right? So it would be a tough thing, right? And from one animal, you can just take out a specific amount, small amount of antibodies, right? So there were two scientists, George Kohler and Caesar Milstein, and you can see their picture also. Kohler and Milstein. Okay, they devised a method for preparing monoclonal antibody. Right. So they prepared a method by which you can develop monoclonal antibodies in the lab, and they did this work in 1975. Okay. Now, what they did in this, they took normal activated antibody producing B cell. Okay. So they took a normal B cell. Okay. And they took the myeloma cell. Right. And by this time, I'm sure you know what is myeloma cell. Right. Myeloma cell is a cancerous plasma cell. Right. What is the role of plasma cell? It secretes antibody, right? So what Kohler and Milstein did, they took a normal B cell, they took a cancerous plasma cell, and they fused the two cell to form a hybrid cell, which is called a hybridoma. Okay, so they took a B cell, normal, and they took a cancer cell. Okay, so this is the myeloma cell. Okay, and then they fused the two cell. Okay, and that led to formation of a hybrid cell. Okay, the hybridoma. Now, what was the purpose of doing this? So there is immortal growth property of myeloma cell. Okay, myeloma is cancerous. Okay, so when it is cancerous, it has immortal growth properties. It will keep multiplying, multiplying, multiplying. So when it will keep multiplying, it will keep secreting antibodies. So you can have a large amount of antibodies. Okay, पहली बात it is secreting antibodies. Another thing, it is not dying. Okay, so it will keep producing antibodies. Okay, so just in a small space you can have a large amount of antibodies. Okay, so the myeloma cell, it provides immortal growth property. Okay, and the B cells, they provide the genetic material for antibodies, right? कौन सी एंटीबॉडी प्रोड्यूस करनी है, ओके, किसके अगेंस्ट करनी है, which epitope is targeted, right? So all that information will come from the normal B cell. Okay, so normal B cell will give the information कि भाई IgM बनानी है against dengue virus, okay, or any other virus, okay, for against a specific epitope. Okay, So B cell gives that information. Myeloma cell का काम है प्रोड्यूस करना एंटीबॉडीज. Just hold on. So students, is this point clear?
Yes, sir.
Okay. So what happens when you mix these two types of cells? It leads to formation of a hybridoma cell which secrete large quantities of monoclonal antibody, and this can be cultured indefinitely, right? Because we are also using myeloma cell. Okay. Is this clear to everyone? Any questions on this?
No, sir.
Okay. Okay, so these two scientists, for their work, okay, they received the Nobel Prize in 1984. Okay, so Kohler and Milstein, they received Nobel Prize for their work for devising a method for producing monoclonal antibodies. Okay, this is what uh, happens, right? So the plasma cells, we are taking the plasma cells. Okay, so these are the normal cells. Okay, so you can see this is the antigen which is injected in a mice. Okay, now this antigen has four different epitopes, but we need a specific monoclonal antibodies, for example, against epitope 1. Okay, now we just take out the cells. Okay, so now you have the B cells which are producing antibodies against epitope 1, 2, 3, and 4. So we have taken the plasma cells, and we have taken the myeloma cells. Okay, and then we have fused the two, right? So now this will have the property of immortal growth property, and it will lead to production of monoclonal antibodies against epitope 1. Okay, and once we have these hybridomas, okay, uh, we can just separate them and use it for our purpose. So whatever purpose we have, we can use. Okay. Okay, now that it was not easy. Okay, one thing is how you form these hybrid cells and how you select these hybrid cells, because in a system there can be different types of hybrid cells or different types of cells. So how you separate your hybrid cell? Okay, so these B cell hybridomas are produced using polyethylene glycol. Okay, so we use PEG or polyethylene glycol. Okay, uh, so, in the presence of polyethylene glycol, the normal cell and the cancerous cells, they are fused. Okay. Now, B cell hybridomas are produced using polyethylene glycol to fuse myeloma cells with B cells from animals that have been immunized with the antigen against which antibody is needed. Okay, so this is important. पहले क्या करोगे? You take an animal, you inject the antibody. No, you inject the antigen. Okay, now as per the natural process, antibodies will be produced against that particular antigen. Okay, B cells will be formed. So you just take those B cells, okay, and fuse them with the myeloma cells, right? So B cell will give the information that antibodies have to be produced against this particular antigen. Okay, and as you know, this hybrid cell will have immortal growth properties coming from the myeloma cell. Okay, and the genetic information for synthesis of antibody, this information will come from the B cell. Okay, so till now things looks easy, right? You just take two cells, put them in polyethylene glycol, and they will fuse, right? But still, there are a lot of problem in practical, or when we actually do it in lab. Not all cells will fuse. Okay, that's another problem, right? So in a culture, in a media, you have so many different cells, and uh, जैसा हम चाहते हैं कि हर सेल हाइब्रिड बन जाए, ऐसा नहीं होगा, right? So there will be a lot of combination, right? For example, unfused myeloma cells. Okay, so you'll have, suppose, unfused myeloma cell जो fuse ही नहीं हुआ, and then you have unfused B cells. Okay, ठीक है. अब जस्ट इमेजिन एक ही सॉल्यूशन के अंदर अनफ्यूज्ड माइलोमा सेल्स हैं, अनफ्यूज्ड बी सेल्स हैं, ठीक है, और बहुत से सेल फ्यूज भी हो गए आपस में. सो देयर कैन बी अ कॉम्बिनेशन की बी सेल और बी सेल ये फ्यूज हो गए. ओके, देयर कैन बी अनदर थिंग कि माइलोमा सेल और माइलोमा सेल फ्यूज हो गए, ठीक है. बट what we need is this thing. We need where B cell and the myeloma cell has fused. Okay, so this is the hybrid cell which we are wanting. Okay, so just imagine एक कल्चर के में आपके पास ये सेल भी है, ये सेल भी है, ये कॉम्बिनेशन भी है, ये कॉम्बिनेशन भी है, और ये कॉम्बिनेशन भी है, and we just want this combination to come out. We just need this combination, right? So there is a problem. Okay, so Kohler and Milstein, they devised a media in which only these hybrid cells will survive. Okay, so what they did, they made a hat medium. Okay, now what does hat means? It's not this hat. Okay, so we are not talking about the hat medium. Okay, so hat medium, it gives a condition for of the procedure which selectively allow the survival and growth of hybridomas only. Okay, so if we grow the cells in hat medium, all the rest of the cells, the B cell, the unfused myeloma cell, the B cell combo, the myeloma cell combo, all will die. Okay, only the hybridomas will survive. So you have to give specific conditions, right? So that only the hybridomas survive. Okay, now using hat media, it's a very common method. Now what happens? Uh, the deficient myeloma cell for one of the nucleotide salvage pathway is taken, okay, making it unable to grow in hat medium. Okay, so हम क्या करते हैं कि हम myeloma cells को लेते हैं, okay, और उनमें से एक जीन हटा देते हैं which produces a specific enzyme. Okay, and that enzyme is required for production of nucleotide. Okay, तो अगर न्यूक्लियोटाइड्स नहीं बनेंगे, so myeloma cells will die. Okay, चाहे वो कैंसेरियस सेल है, but even then it needs all the things so that it can make a new cell. Okay, so हम क्या करते हैं, जो माइलोमा सेल ले रहे हैं, okay, because it will use the nucleotide salvage pathway. Okay, so हम क्या करेंगे, एक एंजाइम उसमें से निकाल देंगे, राइट, सो इट विल नॉट एबल टू यूज this nucleotide salvage pathway, right? So making it unable to grow in hat medium. Okay, so now let's see what this hat medium is composed of. So this hat medium has three main components. One is H, which is hypoxanthine. Okay, A is aminopterin, and T is thymidine. Okay, so this is the full form of HAT: hypoxanthine, aminopterin, and thymidine. And now I will tell what is the role of these three components. Okay, so is it clear till this point? Any question? Okay, is the slide visible?
No, sir.
Okay, let's first have attendance and then we will see how much time is left. Okay, students, let's have a attendance break. Okay, Adish.
Present, sir.
Okay, Atulba,
Present, sir.
Ayushi,
Present, sir.
Sam,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present.
Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswini,
Present, sir.
Namrata,
Present, sir.
Nandini,
Present, sir.
Nehal,
Present, sir.
Rupal, Sahiba,
Present, sir.
okay, Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani, Tanvi, Tridev,
Present, sir.
Vivek, Yukta,
Present, sir.
and Yuvraj.
Present.
Okay, okay. So students, let's get back to this point of hat medium. Okay. So as I said, when we say hat medium, it includes hypoxanthine, aminopterin, and thymidine. Okay. Uh, Now, if we just talk about one of the component, aminopetrin, right? It has a very important role here, right? It was discovered by an Indian. Okay, obviously he was working outside. It's a drug that acts as a powerful folate metabolism inhibitor. Okay, so what aminopetrin does? It inhibits dihydrofolate reductase, okay, with hypoxanthine and thymidine, which are intermediates in DNA synthesis. So I really don't know if you have read the mechanism of DNA synthesis. Uh, have you read that DNA synthesis mechanism in biochemistry,
So
students?
we did.
Okay, so if you have read that, so then it would be easy to understand this, but you don't need to get into that much of detail that we usually do in biochemistry. Okay, you just need to remember that aminopetrin, it inhibits dihydrofolate reductase, okay, which is important in DNA synthesis. Okay, now DNA, now what aminopetrin does is it blocks DNA de novo synthesis. Okay, okay. Now what happens, students? De novo synthesis is the first pathway which is used by a cell to produce DNA. Okay, so now there are two pathways. One is de novo pathway, which is the natural pathway, it occurs. Okay, but in case this de novo pathway is blocked, the cell starts using the second pathway, which is the salvage pathway. Okay, now what we do here is we block the de novo synthesis, okay, and the cell starts using the salvage pathway. Okay, so don't worry if it's not clear, I'll just make it clear in the next few slides. So if mixture of hybridomas and unfused parental cells are placed in hat medium, parental myeloma cell cannot survive. ओके, क्योंकि हमने क्या किया कि हमने माइलोमा सेल में से एक एंजाइम निकाल दिया था, सो वो डीनोवो पाथवे यूज नहीं कर सकता था, ठीक है, और हमने उसमें एमिनोपेट्रिन ऐड किया जिससे वो दूसरा पाथवे भी यूज नहीं कर पाया, राइट? सो माइलोमा सेल विल नॉट सर्वाइव. सो समझ आई बात? जो माइलोमा सेल है, it has immortal growth properties, but we need to kill it because we don't want it. Okay, so we made this hat medium in which the myeloma cells will die. Okay, B cells, you know, every cell has a specific time. Okay, so B cell can survive but do not live for extended period and die out. ठीक है, तो माइलोमा सेल भी किल हो गया, बी सेल तो अपने आप किल हो ही जाएगा बिकॉज इट डज नॉट हैव like it cannot survive much, right? But the hybridomas survive, right? Why? This we need to understand. So we'll understand this process, why only hybridomas survive and why not the B cell. I think it's clear, students, because I think I need to stop now. So if you have any question, you can ask. Okay, so we are standing at a very important, a very interesting point, that we have made a media in which the B cell will obviously die because it does not have the property to keep multiplying, right? But even the cancerous cell will die because we have made some modification in the cell, okay? And we are giving a specific environment, that media, in which it will not survive, right? A B cell If it is alone, it will die. Okay. Myeloma cell, if it is alone, it will die. But when hybridomas are there, right, they will survive. So what happens in this media that they survive, this we are going to discuss in the next class, okay, along with the applications of monoclonal antibodies. So any questions?
Sir, would it be fine if in the next class you could just briefly recap amino pattern?
Oh, sure. Because again, I will discuss, see, when I will now discuss this pathway, it will be very clear what aminopatrin is doing. And I have a slide which shows where aminopatrin blocks the pathway. Okay, so don't worry about that.
Okay, thank you, sir.
Okay, okay. So students, I'm just ending the class now, and in the next class, I think we have tomorrow. So we'll continue with this. Okay.
Thank you.
Okay. Okay.